Okay, great. Well, let me just hide the meeting controls. Okay, fantastic. Well, thanks everyone for joining us today for our special single cell omics panel on single cell proteomics, challenges and opportunities. And so for today, what we'll do is we have three panelists who will be joining us, one whom we may be running a few minutes late. So we'll start with introductions with the two panelists that are here. So myself, I'm Dr. Julia Blasakis. I'm an assistant professor of bioengineering at Rice University. Started just about 2 years ago, and coming from a background of single cell proteomics, in particular single cell Western blot style assay development, especially with a focus on measurement of protein complexes from single cells. So that work was in the Her lab at UC Berkeley for my PhD and postdoc. And so today I'll be telling you kind of in broad strokes about the field of single cell targeted proteomics. Our second panelist who's not here yet is Dr. Sheng Pan, who will be able to share more about the mass spec single cell proteomics kind of blossoming field. And our third panelist is Dr. Ken Chen. Would you like to introduce yourself?
Yes, I can. Hello, everyone. I'm a professor at MD Anderson, bioinformatics, computational biology, systems biology. And I'm a bioinformatician and mostly doing single cell multi-omics data analysis, as well as some of the methods development. And regarding proteomics, I think we've been analyzing, for example, CyTOF data and flow cytometry data on a fairly routine basis, and also increasingly into the spatial omics domain for the different platform, for example, like Codex, IMC, and other stuff. So I'm happy to be here to help answer some of the questions in terms of bioinformatics and methods.
Fantastic. And I can't see currently if Dr. Pan has joined us yet. If I don't hear anything in a minute, I'll just keep going with this portion of the panel.
No, he hasn't. I'm emailing him right now.
Okay, perfect. So we'll kind of have two main sections of slides and presentation. So I'll talk about the targeted proteomics methods and try to give a kind of wide view of the field and its development over the last several years. And then Dr. Pan will provide some background about single cell mass spec advances. Dr. Chen can offer any comments kind of about the challenges and approaches with the bioinformatics methods for some of these types of data sets. And then we'll open it up to Q&A with the audience, really to try to maximize your learning experience so you can figure out what techniques might be well suited to the questions that you work on and how to get started on those techniques here in the GCC institutions and perhaps beyond if others are with us today. So with that, I'll jump right into the targeted single cell proteomic analysis. And so today, I want to first motivate the need for single cell protein analysis. I'll talk about advantages and disadvantages of various single cell protein analysis methods, again, of the targeted variety. And we'll get into what I mean by that versus discovery aspect proteomics shortly. Since my expertise is especially in the single cell Western blotting family of single cell proteomic assays, I'll talk a little bit about the fundamentals of that assay and how it promotes selective detection of proteins in heterogeneous cell populations. And I'll wrap up this section of the panel with some current challenges and opportunities in the field of targeted single cell proteomics. Okay, so as we all know, of course, DNA is transcribed to RNA, which is translated to protein. And a lot of the sort of methods development and advanced analysis has been sort of on the single cell genomics and transcriptomics measurement space. Now, there's a number of reasons why that may not be enough for certain biological questions. So certainly we know that there are many post-translational modifications and events that can occur, including truncation of certain proteins, et cetera, between the RNA and protein portion of this cascade. And interestingly, some of the earliest single-cell multi-omics data sets showed very poor correlation between protein and RNA at the single-cell level. So here each point represents a single cell and the corresponding RNA and protein levels, in this case of MIF, in the cell population. And so overall, it's acknowledged that they're, in part because of the differences between, transcriptional and translational timescales, but certainly because of the different post-translational events that can occur as well, that protein levels may not be equivalent to RNA levels. So if we really want to understand the functional heterogeneity of a cell population, measuring protein may give you a more nuanced picture of what's happening in the cells. I also want to kind of emphasize some classes of molecules, biomolecules that you cannot necessarily measure from an RNA transcript level. So, for example, protein isoforms or complexes would fall into this category, and in particular may be well suited for some of the separation single-cell Western blot style techniques when these particular biomolecules lack selective antibodies for detection. So I'm thinking about here isoforms and protein complexes. So one really great example of a protein isoform that lacks a selective antibody for detection is the HER2 protein in breast cancer. So this this transmembrane protein is amplified in many breast cancers called HER2 positive breast cancer. You may have heard of that type of breast cancer, or you may work on that type of breast cancer. And in HER2 positive breast cancer, enzymatic cleavage can result in this truncated isoform, interestingly, that the therapeutic antibody is not capable of binding to. And so it's actually quite important to be able to measure the levels of this truncated isoform that's generated post-translationally. So it's the protein is produced in its full form and then enzymatically cleaved. So you can't infer this level of this truncated isoform from transcript levels. So being able to measure that sort of heterogeneity is critical, and something that was done at the Her lab when I was still a postdoc there. And so, yeah, being able to identify the subset of cells with this truncated isoform that corresponds with drug resistance is critical, and that's just one example. Another example of classes of molecules that may lack selective antibodies for detection include protein complexes, which was the focus of my work as a postdoc in the Her lab, developing techniques that could measure the relative levels of protein complexes. For example, we focused on the cytoskeleton F-actin complexes comprised of G-actin monomer proteins. And so, again, there's not an antibody that binds only to this molecule, but not to this molecule for the vast majority of protein complexes. And so these types of biomolecules especially lend themselves to some of the separation type techniques. Overall, though, the cellular heterogeneity in protein expression and its non-correlation with RNA requires single cell analysis at the protein level. And so if we take a look at the protein measurement landscape, there's quite a few techniques out there. I won't step through every single one, but I'll talk about kind of in broad scope some of the kind of most influential techniques and ones that you may have access to, and ones that you should be aware of that are just coming into the fray now. So the vast majority of these single cell protein measurements are antibody based for their readout. A small proportion are mass spec based. And so the antibody-based assays are what I'll focus on in my assay today. In my presentation today. Essentially what you can see here is that for all these different types of assays, you're generally dealing with on the order of 10 to 100 proteins detected. Those may be secreted proteins like cytokines, intracellular or membrane proteins, depending on the technique. And you may expect anywhere from sort of hundreds to tens to hundreds of thousands of cells measured as indicated by the throughput here. So we're going to start with the most basic single cell protein assay that pretty much I think everyone in the audience is likely familiar with, but just to get on the same page, all of these antibody-based readouts are essentially utilizing the fact that you can design antibodies that bind selectively to a protein of interest. So in this case here, this red antibody recognizes the purple protein that's on the membrane of the cell, but not the green proteins. So through that selective binding interaction, we have a fluorescent tag that's on the antibody, and so we get fluorescent signal out from this. Generally, these are referred to as immunofluorescence assays, immunocytochemistry when dealing with cells, or immunohistochemistry when dealing with tissues. These types of measurements result in these beautiful images that you're likely familiar with, which increasingly there are great tools such as Cell Profiler or Elastic for being able to retrieve quantitative parameters. But overall, I think we can say that the vast majority of the techniques we'll talk about today are semi-quantitative at best, so you can measure relative levels. Some of them can be brought to absolute quantitation with careful calibration. Okay, so that's sort of the low throughput standard for immunofluorescence. Now, if you want to be able to measure tens of thousands of cells, actually even 10,000 per second, so millions of cells in the case of flow cytometry, would be an assay for you to consider. So flow cytometry uses the same basic immunoassay readout, antibody binding to protein. Now, there are different variations of the immunoassays, some in which the antibody that binds the antigen or protein is the one that's directly labeled. There's also flavors where a secondary antibody binds the primary antibody, and that secondary antibody has multiple fluorophores, and in many cases, multiple secondary antibodies combined for primary, and so you get a sort of signal amplification from that. But overall, these are the types of immunoassays we're dealing with. The cells in this case are passed through, in this case, the flow cytometry fluidics, which narrow down to sort of single file cells passing through this channel here where a laser excites the fluorescence, and then two different detectors record parameters related to size, although not directly correlated, and in many cases poorly correlated. So be careful with that parameter. But as well as the fluorescent signal of the sample. And so from this, you can detect, in this case, up to 14 different markers per cell in some of the kind of most advanced flow cytometers that are available. And certainly we have access to many of those here in the Houston area through flow cytometry core facilities, for example, at MD Anderson, and I believe Baylor College of Medicine as well, possibly others. I don't want to leave anyone out. So, of course, another capability that comes with flow cytometry can be fluorescent activated cell sorting, where cells with particular markers, in this, having your fluorescence signal or not having your fluorescence signal are physically separated and sorted and collected. So that's a capability that comes with some flow cytometers. And then the readout for flow cytometry is perhaps quite familiar from the literature, but essentially these types of pseudo-colored correlation scatter plots to look at correlation of different markers, of course, with red indicating higher levels of expression and blue is lower. Okay, so flow cytometry gets you up to about the 14 markers per cell, which for many questions in current biology, that's helpful, but maybe insufficient. And so over the last 10 years or so, maybe call it 15 at this point, there were advances in the development of mass cytometry, also known as CyTOF, for time of flight analysis. So this is essentially a mass spec readout of a targeted protein, an antibody-based immunoassay. So don't think of it as direct mass spec of the proteins, but mass spec of labels on the antibodies. Okay, so mass cytometry essentially replaces the fluorophores bound to the antibody with metal isotopes. And through the kind of selective analysis of a mass analyzer that can detect differences in the mass to charge ratio, or M/Z, these particular metal isotopes can be carefully and differentially identified more stringently than fluorescence, arguably because you get overlap of fluorescent spectra, which limits typical multiplexing with the standard fluorescence excitation wavelengths and filters to about 3 to 4 colors at a time. Okay, so you can get many more colors in this case, or distinct metal isotope mass overcharge ratios. This is a great review paper for those of you interested in learning more about the fundamentals of CyTOF. But the bottom line is that you can expect to measure about 60 markers per cell. You'll see papers that claim more, but in terms of what is practically achievable with commercially available materials, anticipate around 60. And with a throughput about 100 hundreds of cells per second. So you can certainly get to many thousands of cells with mass cytometry readout. Okay, so this is already pretty exciting, getting up to this kind of 60 marker range. It's part of why mass cytometry and CyTOF have become relatively rapidly adopted, especially in the field of immunophenotyping, especially since the work kind of started out of Gary Nolan's lab at Stanford, and he's primarily interested in immunology. So one of those cases where the technology development occurred to be able to address specific biological questions in immunophenotyping, but has broad applicability elsewhere. Now, for those of you that 60 targets is not enough, there are even other recent advances, as recent as last year, in kind of cyclic assays for measuring proteins, in this case with DNA barcodes. Now, there are multiple types of assays that utilize DNA barcodes. I'll say a little bit more about those later, but in this particular iteration, essentially the antibody against any given target has this cleavable linker and a DNA molecule. So cells are captured in an array in this case, and essentially these are stained in situ. So these are generally going to be fixed and permeabilized cells that are processed on chip. So after allowing the antibodies to bind to either the surface or intracellular targets, the microarray with the cells is interfaced with an array with millions of microbeads. And these microbeads each contain unique oligos that are complementary to specific oligos of the that are bound to the primary antibody conjugate. And essentially, with UV light, the DNAs bound to these antibodies can be released and then collected and hybridized with the DNA-coded beads of what they call the MIST array. So now that's separated from this cell-containing array, and meanwhile, the antibodies can be stripped or removed for another round of staining. And this array here, essentially through a round of what they call decoding, can allow you to identify which proteins were present based on what beads the complementary DNAs are binding to. And so through a round of like 4 successive kind of binding events with the array and kind of collection of hybrid strands, essentially you can identify, for example, bead 1 based on the binding of the 3 different fluorescent colors over the 4 different hybridization and dissociation steps. And so in this particular work, they looked at around 180 proteins from single neurons. They argue that it's possible to substantially increase that multiplexing by continual cycles of this sort of iterative process. That could become cumbersome for some, and I think it really depends on, well, in some sense, the availability of antibodies against your target of interest, and really how much your biological question motivates the need to collect hundreds of different markers. Now, one thing I do want to point out with this sort of method and some of what we've discussed so far, really is we've increased in the multiplexing from flow cytometry to mass cytometry, and then to this sort of molecular collection. It's called SIC-MIST, cyclic. I can't remember what the acronym is, but since they're MIST array, it's a cyclic MIST array assay. All of these higher multiplexed assays start to lose the spatial preservation or spatial information of protein expression that we started with in our kind of most basic immunofluorescence assay. And so that's where a number of different techniques have come in to essentially try to both gain the single cell protein expression information while maintaining localization, and increasing the multiplexing to be able to look at dozens or more targets. So one such technique is imaging mass cytometry. And I understand that this is available in the TMC. I believe it's in the mass cytometry core at MD Anderson, but someone can correct me if that's wrong. For imaging mass cytometry, you use the principle of mass cytometry. So again, these are antibodies labeled with metal isotopes. And so in this case, starting out with your fixed tissue sample, you stain with these antibodies against each of the unique protein targets, generally somewhere around 40 plus protein targets. And essentially, through a laser ablation and collection of the metal isotopes per each square micron of the tissue, you're able to then sort of reconstruct what the localization of the proteins were after collection in the time of flight mass spectrometer. And ultimately, you get very similar images to immunofluorescence and the ability to identify cell subpopulations and using TSNE and different hierarchical clustering type techniques, et cetera. So a nice review of imaging mass cytometry is available. This link here. And in terms of advantages and disadvantages, at least compared to immunofluorescence, so already you've increased multiplexing over 10-fold from the most kind of common immunofluorescence assays. You've eliminated autofluorescence by moving to these metal isotopes with really low crosstalk. The specificity for identifying a particular mass to charge ratio is fantastic in these devices or in these instruments. And so you can get quantitative information with kind of high confidence in your readout. Some disadvantages would be lower resolution compared to standard immunofluorescence, so sort of 1 micron squared per pixel. The expensive equipment and operation costs. So this is not going to be the type of instrument that every person has in their lab at this point. And I think you're going to be relying on core facilities for quite a while in this area. And the data acquisition is quite time consuming. Okay, so with all of that said, if you would like to stick with your immunofluorescence assay, which you have, either microscope in your lab or just down the hallway that you can use, but you still want to preserve your spatial localization and measure many different, dozens of markers. There are a number of cyclic immunofluorescence type assays that have been developed. This is a nice review of many of them. I don't want to leave any techniques out, but I'm sure I will, because this is a very hot area. Essentially, the idea being that, if you have your fluorescent labeled antibodies and stain your cells or tissue of interest and go through some sort of fluorophore inactivation. So this is generally like a chemical quenching, sometimes with pH or specific other chemical reagents to inactivate the fluorophore, and then potentially introduce another round of antibodies. There are also methods with cyclic immunofluorescence that utilize these DNA barcode labeled antibodies. So similar to what I showed earlier with the SICMIST, but one thing I want to mention about DNA barcoded antibodies is that kind of beyond this fluorescence readout, there's increasingly methods that are using DNA barcodes in sequencing-based readouts to quantify protein levels. And so definitely be aware of those as well. I think Ken can speak a little bit about his multi-omics analysis, including site-seq data, which essentially you have a barcoded antibody and you're going to do sequencing of the DNA barcode along with the sequencing of your transcriptome. So it's not just immunofluorescence that these DNA barcoded antibodies are used for. And then, of course, we talked a little bit about the metal labeled antibodies and their readout. Okay, so all this so far has been kind of the types of single cell proteomic methods that are antibody based and not using any sort of other separation step to achieve selectivity. But if we think about the protein isoforms and complexes that I mentioned at the beginning of the talk, of course, there are proteins for which a single antibody binding event isn't going to tell you which protein is actually present, or if your isoform of interest is present. And so that brings us to this single cell Western blotting type techniques for selective detection of proteins in heterogeneous cells. Okay, so for those of you, likely you're familiar with Western blotting. Essentially, this is a technique, workhorse biochemistry technique for separating proteins by their molecular weight in a separation gel, and then transferring those proteins from the gel to a membrane that can be stained with antibodies. Okay, so the single cell Western blot shares some features and some differences, essentially with kind of the workhorse Western blot. So the format is very different from the gel used in standard Western blot. So this is a polyacrylamide gel that's patterned on a standard glass microscope slide. And each microwell here patterned in the surface can accommodate a single cell, and the diameters of those wells can be tuned to the diameter of your cell of interest. And so essentially cells are settled in the microwell array by gravity settling. And then across the entire array, a buffer that lyses the cells and denatures the proteins, at least in the initial single cell Western blot assay format. I'll say more about work that we've developed for selective detection of protein complexes, but in this case, in-well chemical lysis with a denaturing buffer, electrophoretic separation. So this whole thing is submerged in a small electrophoresis chamber, custom designed, and you apply an electric field across the entire array, and proteins migrate into the gel and separate by their size. So as with a standard Western blot, your smaller proteins migrate faster and your larger proteins migrate more slowly. We use a UV mediated covalent capture chemistry to physically immobilize the proteins in the gel, as opposed to completing a transfer step of a Western blot. And so in this step, we use a benzophenone methacrylamide that's polymerized into the gel. This benzophenone is UV activated and creates the covalent bond to any abstractable hydrogen in the protein. So it can immobilize any protein. And so then we're going to do in-gel immunoprobing, essentially diffusing antibodies into our separation gel to measure the relative amounts of each species. And so at the end of one of these assays, you get a readout of something like this. So you have these microwells here. The protein migration was from left to right here. You can see some microwells were empty, so there's no protein signal in the area adjacent to the microwell. But here you can see some proteins bands that are present. And much like a conventional Western blot, if you measure relative levels of the protein species with a densitometry type approach, in this case, it's automated and in MATLAB, and that code's available on GitHub. And I'm happy to talk about that analysis since I helped design that analysis pipeline. But that's kind of the basis of this sort of assay. Now, I mentioned versions of the assay in which you might want to measure protein complexes in particular. So in those types of assays, the actual chemical lysis process is altered, essentially to use complex stabilizing lysis buffers that are used in centrifugation assays for pelleting F-actin complexes. And so in that case, that assay is called SIFTER, and you can look up the paper that I cited a few slides back from 2021 in Nature Communications, where essentially we sift out the protein complexes from the monomers in this microwell. Essentially, the complexes are too large to enter the gel in their complex state, but monomeric proteins in that kind of native lysis buffer and electrophoresis buffer are able to migrate into this gel. And so through a multi-step assay, we're able to physically separate, ultimately dissociate the complexes, migrate them in a distinct region of the gel, and then measure the relative levels of the complex and monomer protein. I'm happy to answer questions about that. I could talk about that assay all day long, but in the interest of time, since we're running low on it, I just want to conclude this portion with some kind of challenges and opportunities in the field. Okay, so first, all of this was antibody-based in terms of the readout of the proteins. And that's a real problem because antibodies are promiscuous at best. So for some references to kind of help you understand the state of the challenges in using antibodies, about 25% of antibodies for epigenetic markers cross-react with non-target proteins. So some representative protein blots showing ways that these types of antibodies can fail, and the corresponding reference here. There's also great work from Emma Lundberg's group showing discrepancies between, and sorry, this is very tiny, but these are N-terminal fusion proteins, C-terminal fusion proteins, and immunofluorescence for a variety of different proteins. And you can see the localizations are not the same at all between these different immunofluorescence and fusion protein assays. And so, we could have a spirited discussion about what's correct in this case, but certainly even since this 2013 paper, there have been other papers showing things like artifacts like transcription factor, leaving the nucleus upon fixation. So it's in some cases a fixation artifact that mislocalizes the protein relative to where it was in the live cell state. So that's a challenge with immunofluorescence and using fixed cells and then antibodies to detect those proteins. Now, in terms of opportunities, I think the sky's the limit here. I think, in particular, I'm quite excited about opportunities in the space of personalized medicine, the readout of specific protein markers that you can't measure at the single cell level, through transcriptomics as biomarkers and prognostic markers, as theranostic markers in terms of deciding on therapies based on each individual patient's, for example, tumor, protein profiling. But certainly, across it's really not just kind of cancer applications, but in general, looking at drug resistance, biomarker identification, certainly in the kind of cancer space, looking at metastasis, the unique cell subpopulation that leaves the main tumor and seeds elsewhere in the body, understanding this tumor and cellular heterogeneity functionally at the level of the protein molecules that carry out the functions of the cell, along with cell-cell interaction and signaling. So one thing I didn't mention that I meant to along the way is some of the suites of methods that are well suited for cytokine detection. So definitely shout out to Rong Fan's lab now at Yale, and his company Isoplexis for kind of making cytokine measurements feasible for the masses. And that's important across a wide range of understanding inflammatory and disease states. So really there's quite a bit of opportunities in the space, and really new methods coming out all the time to facilitate the types of new biological inquiry at the single cell protein level. Okay, so with that, I've run over my time, but I will stop sharing here, and I'll turn it over to Dr. Pan to share some slides.
Okay, can you guys see the slides?
Yeah.
Okay, great. So Julia just gave a fantastic overview on single cell proteomics, especially the target analytical techniques, and also the opportunities and challenges for applying single cell proteomics for research. So I have a few slides that I would like to talk about mass spectrometry-based single-cell proteomics for global profiling of single-cell proteome. So unlike target approach, mass spectrometry-based single-cell proteomic provides a system-wide unbiased profiling of proteins in a single cell. So apparently the major challenge is the extremely small amount of proteins that is available for mass spec analysis. So currently there's no proteome-wide technology that would allow us to do amplification on proteins like we do on DNA or RNA. So the current status of mass spec-based single-cell proteomics is that we are still in a very early stage of technology development. Currently, there are a few platform technology has been introduced. One of the technology is using isobaric label carrier proteome. which was a spike in a single cell sample to enhance the signal intensity. So this carrier proteome is anywhere from 25 to 500 times more abundant than the single cell. So this technique was able to identify more than 2,000 proteins in a single cell. However, recent report also suggests that there are some limitations of this approach. First, there are some concerns about addition of carrier proteome might alter the single cell proteome. And also there's some quantitative measurement issues, and also labeling an extremely small amount of protein in a single cell sample by itself is quite a challenging task. And it also limits the throughput. So recently, Dr. Matthes' group in Germany introduced a DIA-based approach for single cell proteomics. So this approach is based on a high-end, very fancy instrument called TIMS QTOF. So this instrument equipped with trap ion mobility and also 100 Hertz acquisition rate, a very fast instrument. And they use a DIA passive protocol to acquire their single cell data. And again, this technology was able to identify more than 2,000 proteins without using a carrier proteome. And our lab also introduced a spectrum library-based single-cell proteomic platform. This platform is based on a spectrum library that was generated using bulk scale analysis. And we also developed a so-called direct single-cell preparation method, in which we don't transfer our single-cell samples. We did the whole sample process in the LC-MS injection vial, and then directly load it to the mass spec for analysis. And then we use the single cell data to match against the high quality spectrum library that we generate for peptide identification and protein quantification. So the TIMS QTOF method and our method both are spectrum library based, but with a different mechanism. TIMS QTOF method rely on the acquisition speed and the sensitivity of the high-end instrument, and you use MS data for peptide identification, a DIA-based MS data for peptide identification. And we use MS1 data for peptide identification, mainly rely on the hydrophobicity of the peptides based on their sequence and the monoisotopic mass and also isotopic distribution. So our method can use more instrument as long as the instrument is a high resolution and with adequate acquisition speed. So all these three methods will allow us to identify more than 2,000 proteins in a single cell. So I'm going to give you an example how we use the single cell proteomics to identify, to resolve the difference between different cell types and also the heterogeneity between cancer cells. And here it shows the exact ion chromatogram based on spectrum library matching. And you can see that the intensity of the signal and also the complexity of the spectrum increase as the amount of cell available for analysis increase. And the first question we ask is why we are doing single cell analysis. Are there any difference from single cell data from the bulk scale analysis or pseudo bulk scale analysis? So we took our single cell proteomic data, the abundance of the proteins that identify in a single cell, plot against the proteins identified in 5 cells. And you can see that we had quite nice correlation between the data from single cell and the data acquired from 5 cells. And so we increased the number of cells to 10 cells. and the correlation is still there, but not as good. And then we plot the single cell data against the data acquired from 50 cells. Now you can see that they are poorly correlated. So this data suggests that single cell data, at least the abundance of proteins, are not linearly correlated with bulk analysis or pseudo bulk analysis. So to confirm that, we plot the data acquired from 50 cells against the data acquired from 100 cells. Both are considered pseudo bulk sample, and you can see that they are well correlated. So this data underscore the importance of doing single cell protein profiling because they might reveal different proteome landscape that can be masked by bulk scale analysis. And then we use our platform to analyze two cells, normal and cancerous pancreatic epithelial cell. So HPD is the normal pancreas epithelial cell, and then PANC1 is the pancreatic cancer cells. And as you can see here, the first things we found is that the total protein contents that we measure in the protein profiling distinguish the two cell types. The normal cells has a very homogeneous protein abundance, and the cancer cells tend to have a higher total protein abundance, and they're also more heterogeneous. So we did the TSNE visualization, and you can see that again, the blue dots are the normal cells, and they are aggregate together. And again, the cancer cells are more diverse, and there are a few cells representing subpopulations of PANC1 cells, which are distant from the majority of the PANC1 cells. And so this is not a big surprise to us because cancer cells tend to have a higher diversity than normal cells. And then we did the Meccano plot. As you can see, there are significant number of the proteins that show upregulation in cancer cells. And when we analyze those overexpressed proteins that was identified in the PANC1 cells, we found that they were associated with multiple cancer hallmark pathways. And here just shows the pathway analysis that the proteins that are linked to those hallmarks, and they are also heavily interreactive between themselves. So yeah, so I hope this few data, a few slides, the data shown in these few slides can show you that how single cell proteomics can help us to reveal the proteome landscape and detailed information that we cannot obtain from bulk scale analysis. With that, I wanted to show, I wanted to acknowledge our funding agencies, including CPRIT, UT Health, and NIH, and also the Levy Endowment Fund. Thank you.
Thanks for that great overview of the single cell mass spec space. It's super exciting, and one that we're starting to think about how we can advance our separation technologies as preparative steps for single cell mass spec, because I think that's an area that's going to just continue to have huge strides in the coming years. So I see some questions in the Q&A from the audience that I would like us to get to. So someone asked about putting papers in the chat, which I'll coordinate with Dawn to be able to at least share my slides. with all the participants. Hopefully that's doable. And then so from Ryan in the audience, he's asking about the protein complex method that we developed, and if there's kind of any other single cell methods for protein complexes, and if so, what are the benefits and drawbacks of them? Right. So overall, to our knowledge, the sifter assay that we developed is the first for single cell protein separations of complexes, kind of in a higher throughput format. So you can see some capillary electrophoresis work that was attempting to do separations with complexes, but definitely running into some challenges with heating-based dissociation of the complexes in the types of buffers that you need to be able to preserve the cytoskeletal complexes that we were looking at. So in that space, obviously there's other techniques for measuring protein complexes like FRET, proximity ligation assays, at a bulk scale, proximity labeling and mass spec readouts. But some challenges with a lot of the single cell methods are really for one-to-one binding interactions, which if you look at the distribution of protein complexes in the mammalian proteome, the median number of proteins involved in each complex is 4. So that's going to be very challenging to design a FRET or proximity ligation assay for something like that. But with the technique that we developed, we're able to use the kind of size exclusion separation behavior of the polyacrylamide gel to isolate complexes from monomers. That can be tuned for complexes of different sizes. And currently we're actively pursuing a version of the assay that essentially separates protein complexes approximately by size and charge first. And then in a second dimension, forming a dissociating and denaturing protein processing step to then be able to measure the protein subunit compositions in kind of a second dimension separation, so that you can measure stoichiometry and abundance with protein complexes. So if that's something you're interested in, I'm happy to talk more offline about where we're at in that type of assay. Okay, and then I'm not sure that there's a question here. Is the size of the protein its size in the network figure? I'm not sure. This might be for Dr. Pan, I'm not sure.
You mean the physical size of the proteins?
Yeah, they're asking in the network figure whether the protein size is there.
The network analysis is based on the function of the proteins. And so those proteins were identified using shotgun proteomic approach. So for most of the proteins, they identify, especially in the single cell proteomic, only a portion of proteins. And then we use bioinformatic approach to infer the peptide identification to the protein level. I don't know if that answers the question.
Yeah, they say in the legend it had a size in the figure. I'm not sure. I don't recall this personally, but
oh, it's the size of the, yeah.
well, in the interest of time, let's get to more questions. And perhaps the two of you can chat offline to clear up that question. So Junho La is asking about the quality of single cells tested and screened for single cell mass spec, and whether there's kind of a FACS cell sorting procedure done ahead of time, or kind of in general, what sort of sample processing are you doing?
Yeah, so these are cell line samples. So they can be just dispensed and isolated and put into a LC injection vial. And we have developed a direct protocol that allow us to do all the proteomic sample preparation within that injection vial. And then we just take sample directly from the injection vial for LC MS analysis. Yeah, and you assuming that if you do tissue sample, and you might need to have some kind of separation or preparation procedure in the front, like a flow cytometry or single cell sorter instrument like this. So
great. And then maybe one last question. I'm not sure if the audience can see this, but Ray in the messaging to hosts and panelists, Ray says that he's curious about the sensitivity of single cell Western in terms of dynamic range and minimum fold differences that the method can accurately detect. So this is a great question. So the in-gel limit of detection for protein, the protein immunoassay, is about 27,000 molecules. And there's 3 orders of magnitude dynamic range for this sort of fluorescence immunoassay. Now, functionally, this corresponds to more like a median expressed protein in a mammalian cell at the beginning of the assay. So 27,000 molecules at the end of the assay in the gel does not equal you started with 27,000 molecules of that protein, because it's an open microfluidic array. So the proteins are able to diffuse out of the top during the assay. So it's kind of a race against time there. But with that said, we can get through 3 orders of magnitude and cover the top half of the proteome. Now, in terms of fold differences, we've done some characterization in the past on biological versus technical variation. And that showed that any variation kind of above about 25%, CV of 20, around 25, don't quote me on that exact number, around 25% CV was where we could distinguish biological from technical variation. So that means that I can't give you an exact fold change number off the top of my head, but if you take that into account with the kind of 3 orders of magnitude dynamic range, that means that there will be some fold differences that are distinguishable and others that are not, depending on the kind of expression level of the proteins. So I hope that helps answer that question. And so with that, we are just about out of time for our panel today. I want to thank everyone for attending and for the great questions. If there are remaining questions, please feel free to reach out to any of us on the panel. We're happy to try to help guide you to where you can get started on some of these techniques. Definitely over here at Rice, we're eager to help get folks started in single cell Western blotting or sifter type assays. So please reach out if you have isoform or complex of interest that you can't measure with other techniques, and we can help you get started with that sort of assay. Any closing comments from you, Dr. Pan?
Yeah, I think thank you for inviting me to attend this very nice panel discussion. And yeah, I think this is a great technology that is still in the early stage. So a lot of work need to be done. And so that is why it's so excited about the things happening in this field.
Great. And of course, thank you to the Gulf Coast Consortium, the GCC, for organizing and their support of the panel, as well as the single cell omics clusters activities year round. So stay tuned. We have, of course, the annual symposium in October. So if you're interested in learning more about single cell analysis, please be on the lookout for the call for abstracts and registration info for that as well. So with that, have a great afternoon, everyone, and thanks for joining.